Hello, and welcome to the CldU IdU staining protocol. Today our video will begin with Step 3, rehydration, permeabilization, and antigen retrieval. Begin by loading your slides into the carrier. Once your slides are loaded, place them into a xylene bath for 5 minutes. This will be followed by a second xylene bath, again for 5 minutes. Continue to rehydrate your slides by moving them into ethanol solution, starting with 100% solution and finishing at 50%. You will then complete slide rehydration by placing your slides for 5 minutes into 1x PBS. To permeabilize cell membranes, immerse your slides in 0.2% Triton X solution for 5 minutes. Prepare your slides for microwave tissue antigen retrieval by placing them into a glass beaker with sufficient volume of 0.01 molar sodium citrate buffer, so that the solution covers 5 centimeters above the top of the slides to account for evaporative loss.
Microwave slides until the solution is boiling. This should take about 3 to 10 minutes at full power. Check that slides are boiling. Reduce microwave power between 10 and 80% and continue slowly boiling the slides for an additional 20 minutes. After boiling, place the beaker containing the slides on a bench top to allow them to slowly cool for approximately 2 hours until they've reached room temperature. Confirm that slides are at room temperature. Immerse the slides in 1.5 natural hydrochloric acid for 40 minutes at room temperature.
Remove your slides from the hydrochloric acid and place them for 5 minutes into a 1x PBS wash. This will be followed by a second 1x PBS wash.
Remove your slides from the 1x PBS and assess solution. With a liquid blocking wax pen, circle each tissue section and replace the slide back into the PBS.
Make a humid incubation chamber by placing a wet paper towel at the bottom of an airtight container.
Make a blocking solution of 10% docking serum, diluted in 1x PBS. It is important to entirely cover each tissue section with the solution. For tissue sections that are typical, about 1 by 1.5 centimeters, a volume of 50 microliters of blocking solution is sufficient.
Prepare the primary IdU antibody by diluting mouse anti-BRDU antisera in 5% donkey serum at a concentration of 1 to 250.
Pipette the primary solution onto each slide section.
Place your slides into a refrigerator at 4 degrees Celsius for an overnight incubation. Prepare a high stringency wash. TBST works well.
Orienting slides back to back, place the sides in about 35 to 40 milliliters of TBST solution. Place the tubes in a shaking bacterial culture incubator at 37 degrees Celsius. Allow them to shake for 20 minutes at 225 RPM. Now load your slides into a carrier. Place your slides in 1x PBS for a 5-minute PBS wash.
This will be followed by a second 1x PBS wash for 5 minutes. Prepare the primary CldU antibody solution by diluting rat anti-BRDU in 5% donkey serum at a concentration of 1 to 250. Tap slides on a paper towel and then resection with a wax pen. Use a pipette to cover each tissue section with primary CldU solution. Use about 50 microliters of solution to make sure that every tissue section is completely covered. Place your slides in a 4 degrees Celsius refrigerator for an overnight incubation. Place your slides in a carrier and then place them in a 1x PBS wash for 5 minutes. Place your slides into a second 1x PBS wash for 5 minutes. Prepare secondary antibodies by diluting Cy2 donkey anti-rat and Cy5 donkey anti-mouse antibodies in 5% donkey serum. DAPI will also be used for nuclear staining. Completely cover tissue sections with secondary antibodies. 50 microliters of solution should be sufficient to completely cover your tissue. Place the rehydration chamber in a dark place at room temperature for a 1-hour incubation. Place your slides in a carrier and then place them in a 1x PBS wash for 5 minutes. Place your slides in a second 1x PBS wash for 5 minutes. Tap off excess PBS solution on a paper towel. Then take Prolong Gold anti-fade mounting media and place a small drop of it on each tissue section. Take a cover slip and place it gently on top of your slide sections. Use the paper towel as a press to push out any excess air bubbles that may have been trapped between your slide sections and the cover slip. Finally, use a pipette tip to push out any excess air bubbles that may remain. Image your tissue of interest using a fluorescent microscope equipped with a high-energy light source and multiple fluorescent channels. To quantify results, begin by counting the number of DAPI-positive cells. Count the number of CldU positive cells using the Cy2 channel image. Count the number of IdU positive cells using the Cy5 channel image. Finally, count double positively labeled cells by flipping between the Cy2 and Cy5 channel image files.